Hi, my name is Brianna and I'm a tutor with Chegg, and today I want to talk to you all about nucleic acids. So nucleic acids are DNA and RNA, and that DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. So those are two differences in the two types of nucleic acids that you can have. And the reasons why they're called nucleic acids is because way back when we were just figuring out what all happens in cells and what is genetic information and all of those things, some investigators noticed, then a lot of info is inside of the nucleus. And there's really important stuff inside the nucleus. So they determined that whatever it is in there, they're going to call it nucleic acid. And then when we started figuring out that there are two different types for DNA and RNA, then that's when the breakup happened. But they're still very similar, so they're still within the same group. So let's talk about what the structure looks for nucleic acids first, and then we'll go into more details about the two different types. So for when you have a nucleic acid, that's what's called a polymer, and polymers are made of a bunch of monomers. And the monomer for nucleic acid are nucleotides. So there's a bunch of nucleotides in every nucleic acid. So tons of these continuously. So if you had an RNA, you're going to have a ribonucleic or a ribose sugar, and that it will just look like this guy. So this is a ribose, but if you have a deoxyribonucleic acid, a DNA, you're going to have deoxyribose. And deoxyribose has an oxygen right here. So there's going to be an oxygen in between these two carbons, and that's what's going to be holding it together. So that's the difference for the sugars between the two different nucleic acids that you could have. But every single nucleic acid will have a nitrogen-containing base coming off of it, and it'll look like this purple guy. And then there's a phosphate group coming off of the other side, and it'll always be a PO4. So think of it like the sugar in the PO4 will always be the backbone for your nucleic acids. And so a DNA is a double helix, and it has the sugar phosphate backbone all along the back parts of this, okay? And then the nucleic acid is what's connecting it. And when I say double helix, what that means is it's like a twisting ladder. Think of it like a rounded staircase sort of thing that you see in cool lighthouses or something like that. That's what your DNA looks like, is it's just wrapped up. And the reason why it's so crumbled and everything is because it's just so much information that if you had it just straightened out, it couldn't fit inside of our nucleuses. That's just such a small space for all the DNA information that we'd have. So here are the base pairs, and these are the base pairs of your nucleotide, or your different nucleic, what word am I looking for? So these are your 4 bases. Sorry, everyone has to correct themselves sometimes. So it'll be the bases that are attaching to each other. And those, for DNA, there's just 2 different pairings that you can have. They never cross pair. So cytosine and guanine are always going to pair with each other. And this is what the nitrogen-containing base would look like. So what's attached right here to your nucleotide will be one of these bases. So you have your cytosine, and that would pair with the guanine, and that would be on the opposite edge of the DNA. So the cytosine and guanine are going to pair with each other. And then you're going to have thymine and adenine pair with each other as well. And they can go in any order, and the order that they go in is what kind of genetic information is being relayed to the cell or to wherever, just saying like, hey, this is what's being determined right now. We're going to be able to transcribe this, whatever the DNA needs to do. All the genetic information is determined depending on what order the base pairs are in, which is pretty cool that you can have so much differing information based off of 4 nitrogen bases. And now, let's talk about the difference between DNA and RNA. We already said that the difference would be the sugar, and the other difference is the type of base pair. So there still is an adenine, there's a guanine and a cytosine pairing, but thymine is not in RNA. So RNA is a single strand. It's not a double helix. It's not a double strand like DNA is. RNA just has a single strand. And RNA is used, do this real quick. So RNA is transcribed from DNA. So it's transcribed from DNA and then it's translated into proteins. And the translation is based on the 3 codon pair, and that tells that determines what kind of proteins are going to be created depending on the order of the pairs of the, not pairs, the order of the nitrogen pairs that you are that you have in your RNA. And that's what's being determined for the proteins. So you still have your guanine, you still have your adenine, you'll still have cytosine, but the difference is you won't have thymine, and your thymine is getting replaced by uracil, and the uracil is the U. Okay, so that's the one difference, or one of the differences between RNA and DNA, as uracil is there instead. And yeah, so those are like the big things for nucleic acids. If you have any questions for me, please let me know. I would love to help you out. And I hope you have a great rest of your semester. Bye.